Most aquarists keep a jar of these on the shelf next to their fish food. Some aquarists keep a jar of these instead of fish food. The distinction, it turns out, is more philosophical than practical. This is Ceriodaphnia, and Ceriodaphnia has opinions about being called a snack. Let us formally introduce our new friend. Genus Ceriodaphnia. Class Branchipoda. Phylum Arthropoda, the same phylum as shrimp. It is technically a crustacean, a distant relative of every shrimp, crab, and copepod we've met on this channel. The same blueprint, the same basic body plan. Scaled with considerable ambition down to less than 1 millimeter. Those antennae are not antennae in a sensory sense, they are oars. Ceriodaphnia swims by rowing. A full power stroke, a recovery, a power stroke again. Dozens of times per minute, perpetually. Ceriodaphnia does not coast. One eye, compound, a single dark spot fixed at the front of the head, and disproportionately large. Larger relative to body size than almost anything else in a normal aquarium. There is no reasonable explanation for why this animal is this small and needs an eye this conspicuous, and yet it is. There are 5 pairs of limbs along the underside. They are not legs, they're fans, fanning inward and outward in a coordinated rhythm, generating a current, sieving particles from the water column, phytoplankton, bacteria, suspended detritus. Ceriodaphnia is, in the most literal sense, a living filter. And like every arthropod on this channel, it periodically discards that entire outer body and grows a new one. Ceriodaphnia molts. Of course it does. They all molt. This is apparently one thing evolution decided was non-negotiable. Under favorable conditions, a female Ceriodaphnia has made a reproductive decision that is either deeply pragmatic or quietly radical, depending on your perspective. She does not need a mate. She reproduces by parthenogenesis, from the Greek parthenos, meaning virgin, and genesis, meaning origin. Her eggs are unfertilized. They develop anyway. The offspring are her genetic replicas, copied with faithful efficiency of a photocopier that has never once jammed. Something I know nothing about in the teaching profession. Generation time is only 3 days at optimal temperature. A single female produces 5 to 15 offspring per clutch. Then 3 days later, she does it again and again. This is not reproduction, this is population policy. A culture can go from a modest founding colony to thousands of individuals in under 2 weeks. The math here is not subtle. It is the very kind of math that makes ecologists nervous and fish keepers happy. The embryos develop inside the carapace. Sheltered, provisioned, protected. There is no larval stage, no nopli, drifting helplessly through the water column, no intermediate form that requires a separate food source or a separate prayer. They arrive as functional miniature adults. This channel has now noted the absence of larval stages in shrimp, snails, and now Ceriodaphnia. The pattern is not a coincidence. Direct development is an investment in survival. Larvae are expensive. Ceriodaphnia has decided not to bother. Parthenogenesis is efficient. It is fast. It is scalable. It has, however, one structural limitation that Ceriodaphnia is well aware of. Every female is a clone of her mother. Who was a clone of her mother? Who was also a clone? The population has exactly as much genetic variation as a single individual. This is fine until it isn't. When food runs low, when the population outgrows the jar, when the days shorten and the temperatures drop, Ceriodaphnia reads these signals, and Ceriodaphnia has a contingency. She begins producing males, which she has not needed until this moment. The males fertilize the eggs, and those eggs are then enclosed in a structure called the Ephippium, from the Greek for saddle, because it is shaped exactly like a saddle and sits on the female's back. And whoever named it simply described what they saw, and honestly, fair enough. The Ephippium does not sink, it floats, kept buoyant by a pocket of gas-filled tissue, drifting at the surface until it washes the shore or lodges in sediment at the water's edge. It hardens as it travels. It becomes essentially a waterproof, sunproof, time-proof capsule, built to survive complete desiccation, ultraviolet radiation, and years of waiting for conditions worth waking up for. The Ephippium is not a failure state, it is the design, the entire sexual reproductive apparatus. The males, the mating, the fertilization, the whole cumbersome procedure. It exists for one purpose: to produce an indestructible egg with a reshuffled genome that can outlast catastrophe. When the rains return, when the pond refills, when the aquarist adds fresh water and algae to a jar they forgot for about 6 months, the Ephippium hatches. The population begins again, genetically diverse, ready for whatever ended the last one. A Ceriodaphnia culture is not just livestock. It is a readout. A behavior of the population tells you in real time what is happening with the water chemistry inside that jar. You simply have to know what you're looking for. Surface clustering. This is the first signal. When dissolved oxygen drops, Ceriodaphnia, like the ram's horn snails, like virtually every other aquatic organism with a choice, moves toward the air-water interface. They are not socializing, they're suffocating, politely, at the surface. A healthy Ceriodaphnia is already translucent, brownish-orange, that baseline tint that itself is a small dose of hemoglobin, present even in decent water. Drop the oxygen further, though, and the color deepens towards a true red. The mechanism is the same iron-based oxygen-binding molecule found in your blood, documented across the cladoceran family and produced convergently by ram's horn snails, some fish, and now apparently by an animal smaller than a grain of rice. Ceriodaphnia simply starts the conversation already partway there. A clear jar with slow, lethargic individuals near the bottom has a different problem. Not oxygen, but food. The algae is gone, the culture has eaten itself out of house and home and is now in a population crash. A sexual reproduction event, or perhaps both. This is not a crisis, this is information. Feed it or split it. Here's the part that elevates this organism beyond the hobbyist curiosity. Ceriodaphnia dubia is not just a bioindicator in your tank. It is one of 3 cladocerans formally adopted by the United States Environmental Protection Agency for regulatory water quality testing, standing alongside Daphnia magna and Daphnia pullix as species used nationwide to evaluate wastewater treatment effluent before it reaches a river. When a treatment plant needs to know whether its effluent is safe to discharge, regulators do not always reach for a chemical assay. They reach for this animal. Its short life cycle, a full reproductive test in 7 to 10 days versus 3 full weeks for Daphnia magna, and its outsized sensitivity to contaminants make it an early warning system that a spreadsheet cannot replicate. This test is not done just to ask whether the animal survives, it asks whether it reproduces normally. A chemical that does not kill Ceriodaphnia outright, but quietly suppresses its offspring count, is still a chemical doing damage, and Ceriodaphnia will tell you that, days before a fish would show any symptoms at all. Active individuals distributed throughout the water column with heart rates up and antennae in full stroke, that is a healthy culture. That is also, for what it's worth, the exact same readout a federal regulator would be looking for in a compliance lab 3 states away. We have spent previous episodes building a food web, the producers, the decomposers, but what we have not formally addressed is the middle, the layer that connects the algae to the fish, the detritus to the shrimp, the sunlight on the surface to the animal that eventually eats it. Ceriodaphnia eats phytoplankton. It eats bacteria. It eats suspended organic particles, the fine particulate organic matter that drifts through the water column after larger detritus breaks down. It consumes primary production and microbial biomass and packages it efficiently into animal tissue. Every fish that eats Ceriodaphnia is eating concentrated algae, compressed bacteria, processed detritus. The Ceriodaphnia has done the conversion work, turning diffuse, low-quality food sources into dense, high-quality prey. This is not fish food. This is a trophic service. And because Ceriodaphnia reproduces at the rate that it does, it can sustain predation pressure that would collapse almost any other prey population. It is not defenseless. It is demographically invincible. It is difficult to out-eat a population that reproduces itself every 3 days. In the wild, this is how lakes stay clear. Cladoceran zooplankton, Ceriodaphnia, Daphnia, and their relatives filter the water column. Remove them through overfishing, through pesticide runoff, through any disruption to their population, and the algae blooms. The lake goes green, the light disappears, the ecosystem collapses from the middle out. The smallest animals in the lake were holding it together. This jar is a microcosm. Not as a figure of speech, literally, a small world. Inside, there's a primary producer, a primary consumer, a nutrient cycle, a population undergoing boom and bust dynamics, and, if you let it run long enough, a decomposer community establishing itself in the sediment. This is a functioning ecosystem. It holds approximately 1 liter. The algae is the producer. It photosynthesizes. It grows. Ceriodaphnia is the consumer. It filters. It reproduces. Together they form the simplest possible food web. 2 species, 1 direction of energy flow, 1 ecological relationship. Everything else is a variation on this theme. Stop feeding the algae and watch what happens. The Ceriodaphnia clear the water. Green water becomes clear water in 1 to 2 days, depending on the population density, of course. Then the Ceriodaphnia slow. Then they cluster at the surface. Then they crash, or switch modes, or both. This is boom and bust population dynamics. This is predator and prey cycling. In a jar, on your shelf. Oxygen is the invisible constraint on all of this. Ceriodaphnia can tolerate brief, moderate hypoxia by raising its hemoglobin, but not indefinitely, and not at 0. Gentle aeration can help keep the water column mixed, the oxygen available, and the algae suspended where the Ceriodaphnia can reach it. Without it, the ecosystem stratifies. The algae floats, the Ceriodaphnia struggles, and nothing works. Look at the bottom of the jar. The sediment layer is a record. Molted exoskeletons, because Ceriodaphnia molts, because everything molts, because apparently we have made peace with this. Dead individuals, fecal pellets, detritus from incomplete digestion, bacteria colonizing all of it, a decomposer community establishing in the sediment of a mason jar. You did not mean to build all of this. You were just trying to feed your fish. But you built a food web. You built a nutrient cycle. You built a population that reads water chemistry and signals it back to you through its behavior. You built an ecosystem, and you probably paid about $4 for the jar, inflation notwithstanding. Inside an animal, smaller than a grain of rice, there is a heart. It beats approximately 200 times per minute. There's a circulatory system. There are muscles driving it. There are eyes watching, antennae rowing, embryos developing in a sheltered pouch, a population ready to double in 3 days, a genome that has been cloned so many times and so faithfully that it will switch to sexual reproduction the moment it senses catastrophe approaching. Ceriodaphnia is a suspension feeder and a primary consumer, a trophic link and a biological filter, a bioindicator, a model organism, a convergent hemoglobin producer, and a textbook demonstration of phenotypic plasticity, R selection, and the adaptive logic of reproductive flexibility. It clears algae from lakes. It feeds juvenile fish in ways that no pellet can fully replicate. It responds to hypoxia by turning red and to starvation by building an indestructible egg. It has been doing all of this in ponds and ditches and drainage channels for over 400 million years. It is also, incidentally, fish food.